Imagine you're photocopying a document that already has typos. Each copy adds more mistakes until the text becomes unreadable. Early life faced a similar nightmare. Every time a primitive molecule copied itself, it introduced dozens of errors. How did these first replicators avoid a mutational meltdown? Keeping enough correct information to evolve complexity instead of collapsing into chaotic gibberish? What exactly is the error threshold? It's the tipping point where the replication error rate outpaces natural selection's ability to preserve useful sequences. Below this threshold, functional molecules outcompete mutants. Above it, harmful mutations accumulate faster than they can be purged, leading the entire population toward extinction. How might early replicators have lowered their personal error rates? Even simple RNA enzymes, ribozymes, can fold into structures that subtly favor correct base pairing. Metal ions, like magnesium in volcanic pools, stabilize these folds, reducing misincorporations. Think of it like adding a proofreading assistant to your photocopier, so fewer letters get mangled. Could compartmentalization serve as an evolutionary life raft? Yes. Tiny lipid bubbles, or mineral pores, in hydrothermal vents trap related copies together, concentrating resources and buffering against outside mutants. In such microenvironments, a handful of high-fidelity templates can dominate, much like a small study group improving their test scores by sharing notes in a quiet room. Do modern viruses teach us anything about the edge of collapse? RNA viruses like influenza or HIV live dangerously close to their error thresholds. Researchers exploit this by flooding them with mutagenic drugs, pushing them over the edge into lethal mutagenesis, where accumulated errors render the virus non-viable. What about cooperative networks? Can they rescue information? IEN's hypercycle model shows cascading autocatalytic loops, where molecule A helps make B, B helps make C, and so on, forming a protective circle. This network structure amplifies fidelity. Errors in one link can be corrected by the cycle's mutual support, like teammates catching each other's typos in a group project. How do modern cells keep mutation meltdown at bay? Today's life adds layers of error correction. DNA polymerases proofread and excise mispaired bases, mismatch repair systems patrol the genome, and chaperone proteins ensure proper folding. It's akin to having multiple spell checkers running simultaneously, each catching different types of mistakes. Why does this dilemma matter for synthetic biology and exobiology? When designing artificial cells or searching for life on other worlds, we must ensure replicators have both sufficient copying fidelity and mechanisms, compartments, networks, proofreading to avoid meltdown. Understanding how primordial life threaded this razor's edge guides us in creating robust synthetic organisms and recognizing life's signatures beyond Earth. Key takeaway: life's origin demanded a fine balance, enough mutation to innovate, yet enough fidelity to preserve function. By folding into protective shapes, hiding in tiny compartments, cooperating in networks, and gradually evolving error-correcting tricks, early replicators stayed just below their mutational breaking point, laying the groundwork for the vibrant, diverse biosphere we see today. The error threshold marks the maximum mutation rate beyond which genetic information can no longer be reliably maintained in a replicating population. If early life exceeded this threshold, deleterious mutations would accumulate inexorably, leading to mutational meltdown and extinction. Yet primitive replicators somehow kept mutation rates below this critical value long enough to evolve repair and proofreading mechanisms. Understanding how they averted meltdown sheds light on the origins of genomic fidelity. Example, RNA viruses, which operate near their error threshold, deploy R-independent RNA polymerases with modest proofreading to avoid catastrophic mutation accumulation. Analogy: it is like walking on a tightrope. One misstep too many sends you crashing, so you learn balance techniques to stay safely within the tolerance limit. The mutation rate is the probability that a nucleotide is incorrectly copied during replication, while the error threshold is the upper bound on that rate compatible with information retention. Below the threshold, selection can purge most harmful mutations and preserve the master sequence. Above it, the population's genetic makeup drifts randomly and the master sequence is lost. Distinguishing these concepts clarifies why life requires both replication fidelity and selective pressure. Example: In theoretical quasi-species models, a single beneficial genotype persists only if the product of genome length and per base mutation probability stays below unity. Analogy: it is like sending messages through a noisy channel. If the error rate is too high, even the best error-correcting code fails to recover the original text. Early replicators formed Quasispecies, clouds of related genotypes centered on a high-fitness sequence. Genetic drift within this cloud can push the population past the error threshold if mutation pressure overwhelms selection. However, robust fitness peaks and mutationally buffered networks helped maintain integrity by allowing neutral or compensatory mutations. These dynamics explain how primitive molecules could survive despite high mutation loads. Example: computer simulations of RNA replicators show that populations avoid meltdown by evolving flatter fitness landscapes where many genotypes confer similar fitness. Analogy: it is like a troop of tightrope walkers who support each other's balance, so if one wobbles, the group keeps the line stable. Natural selection acts as a sieve, favoring replicators with higher fidelity or robustness to errors. Even rudimentary repair pathways, such as template direction cleavage of mismatches, reduced effective mutation rates. Over time, these mechanisms co-evolved with genome replication to push the population safely below the error threshold. The interplay of selection and repair thus established the foundations of modern DNA-based life. The primitive ribozymes with exonuclease activity improve replication accuracy by excising incorrectly paired nucleotides. Analogy: it is like proofreading your own typing as you write, catching most typos before they snowball into unreadable text. Causing large population sizes buffer against drift, making it easier for selection to eliminate deleterious mutations before they fix. Conversely, bottlenecks, such as harsh environmental shifts, shrink populations and can momentarily push them toward meltdown. Early life likely experienced cycles of expansion and contraction, tuning population size to maintain fidelity. Balancing these dynamics helped primitive organisms stay on the safe side of the error threshold. For example, seasonal booms and busts of microbial mats and tide pools maintain effective population sizes large enough to avoid meltdown despite harsh conditions. Analogy: it is like a company retaining enough staff diversity that a few departures don't cripple operations during busy seasons. Ratchet describes irreversible accumulation of harmful mutations in small asexual populations, effectively ratcheting up genome decay. The ratchet clicks faster when mutation rates approach the error threshold, accelerating meltdown. Nonlinear interactions between mutation and selection create sharp transitions from viability to collapse. Early life had to develop counter-ratcheting strategies before this irreversible decay took hold. Example: laboratory studies of yeast lacking recombination show rapid fitness loss unless sexual cycles periodically reset the ratchet. Analogy: it is like a ratchet wrench that only tightens. Without a mechanism to loosen it, the system grinds to a halt under constant torque. Avoiding mutational meltdown was crucial for the emergence of complex biochemistry and large genomes. Viruses today hover near their error threshold, balancing rapid evolution with genomic integrity. Studying how early life managed this trade-off informs antiviral strategies that push viral populations over their threshold. Insights into these dynamics also guide synthetic biology efforts to design stable, minimal genomes. Prominent example: ribavirin's antiviral action increases mutation rates in RNA viruses, deliberately driving them past their error threshold. Analogy: it is like adding noise to a radio signal until the broadcast becomes unintelligible. Reconstructing early replicator dynamics requires integrating chemical kinetics, replication fidelity, and selection in variable environments, a formidable modeling challenge. Experimentally mimicking primitive replication systems in vitro often leads to rapid error accumulation unless modern enzymes are present. Disentangling the contributions of population dynamics versus inherent molecular robustness remains difficult. Addressing these challenges demands interdisciplinary approaches, bridging theory and laboratory evolution. Example: attempts to evolve error-prone polymerases in the lab often select for unintended high-fidelity variants that confound meltdown studies. Analogy: it is like trying to build a prototype car engine that runs on mud. Practical tests routinely default to gasoline engine components. Advances in microfluidic evolution platforms will allow precise control of mutation rates and population sizes to probe error threshold phenomena. Synthetic minimal cells with tunable replication fidelity offer testbeds for studying mutational meltdown. Theoretical work on error-correcting codes in biology may reveal new principles underpinning genome stability. Ultimately, unraveling the error threshold dilemma illuminates how life first achieved the delicate balance between innovation and preservation. For example, novel droplet-based reactors can cycle replication compartments through bottlenecks to observe real-time bust and boom fidelity dynamics. Analogy: it is like using a high-speed camera to capture each frame of a collapsing house of cards, revealing the exact moment when structural integrity fails